Last year, the FDA approved Elacestrant, or Orserdu, to treat metastatic estrogen receptor-positive breast cancer with mutations in the estrogen receptor gene, which is called ESR1. In this episode, we'll be referring to this as ESR1 mutations. This approval was a long time coming for Komen Scholar and breast cancer researcher Dr. Donald McDonnell, whose lab discovered Elacestrant.
It's a great story, and it's a story that really highlights the ingenuity of some of the trainees in my lab. But the story didn't start a few years ago. It actually started about 25 years ago when I was interested in why is it that cancer cells become resistant to, at that time, the best endocrine therapy was tamoxifen. And we thought at the time it was just because tamoxifen was a good drug, but not a great drug. Let's go make better estrogen receptor inhibitors. And one after the other, the field was making these estrogen receptor inhibitors, testing them in tamoxifen-resistant disease, and they were all failing. And in fact, my last count, when I wrote an article on this recently, there were 9 drugs that were evaluated for efficacy in patients who had failed tamoxifen. And so rather than just wanting to do the same thing over and over again, we decided to actually see, was there another way that we could actually inhibit the estrogen receptor in tumors? And the idea was to develop small molecules that would actually cause degradation of the estrogen receptor and then test them. I won't go through all how we got to them and things like that. It was a lovely science project published in a fantastic paper by the two of them in about the year 2000. And they found a drug. And the drug bound to the receptor, twisted the receptor into a shape that led the cell to believe that it was a denatured protein, and the protein was then thrown into the cell's trash can, and the estrogen receptor was gone.
After the initial discovery of drugs that could get rid of the estrogen receptor in breast cancer cells, which are also called SERDs, Dr. McDonnell's lab ran into some roadblocks. They were working with a few companies to move this drug into clinical trials, which is how we test new drugs for effectiveness and safety. These companies lost interest in developing this type of drug, and eventually the group needed to start from scratch with brand new molecules. They ended up with a molecule that was originally unsuccessful for treating hot flashes, which intrigued Dr. McDonnell's research team.
So what they found was, in a clinical study, was that as you increase the doses of this hot flash, of this treatment, you actually increased the number of hot flashes, not decrease them. And so Suzanne Wardell and Eric Nelson, who are 2 trainees in my lab, basically love weird pharmacology. Okay. And so they got their hands on some of this drug, and within 2 weeks, they had figured out the mechanism. And it turns out the very mechanism that was probably responsible for the failure in hot flashes was the very reason that it worked in breast cancer. Because what it did was, it did the same thing as our original drug. It bound to the receptor, it twisted the receptor into a conformation, and that was denatured, and the cell threw it into the trash can and was gone.
Finally, by 2017, Dr. McDonnell handed the new drug to a pharmaceutical company who started a phase 3 clinical trial of the drug for breast cancer in 2019. And this trial was called the Emerald trial.